Hello, everyone. Thank you for those joining us both virtually and in person. Welcome to our first lecture from our Heart Month lecture series. It's something we do every year now. So over the next 3 Fridays, including today, we'll have cardiovascular focused research presented in recognition of February being Heart Month to raise awareness for heart disease. We're very fortunate today to have Dr. Nan Wang join us from Stanford University. And we're incredibly grateful because since she wasn't able to travel here due to COVID reasons. She got up so early, I think it's 6 o'clock out there in California now to join us for a presentation. So we're very thankful for that. So I've gotten to know Dr. Wang over the last several years on the executive committee for the International Society for Applied Cardiovascular Biology, where we both serve, and she serves as the membership chair. And she's assistant professor in the Department of Cardiothoracic Surgery at Stanford, and a PI in the Veterans Affairs Palo Alto healthcare system. Prior to Stanford, she completed her BS in chemical engineering from MIT and a PhD from UC Berkeley, UC San Francisco Joint Program of Bioengineering. She was also a postdoc in the Division of Cardiovascular Medicine at Stanford University. And her lab investigates interactions between stem cells and extracellular matrix environment for engineered cardiovascular tissues, with a focus on treating cardiovascular and musculoskeletal diseases. She's authored over 90 publications and patents, and including reports in Nature Medicine, PNAS, and Nano Letters. She has received numerous honors, including funding from the NIH, a K99R00 Career Development Award, and other significant funding from the NIH, NSF, Department of Defense, Department of Veterans Affairs, and the AHA. So we're very fortunate to have you here today. Looking forward to the presentation. Thanks, Nan.
Thank you very much. Well, let me share my screen with you all. Great. Well, thank you again for this kind introduction and invitation to speak. I know that this is a really special month for you all, being Heart Month. And now when we think about the heart actually is composed of many different cell types and structures in the heart. And I know that some of the later seminar speakers will be talking about the actual heart muscle itself. And so for this talk today, I wanted to focus on another important element of the heart, which is the vasculature. And so today I'll be talking about how the extracellular matrix cues modulate the vasculature and how we can use that for creating vascularization. And so the blood vessels in your heart and throughout your body play a really important role in maintaining vascular homeostasis. Basically, the endothelial cells are the cells that line that inner layer of the blood vessels, and they are the cells that actually interact physically with the blood and all the circulating blood elements. The endothelial cells we think of as being kind of like cellular Teflon, because they are what prevents things like lipids and proteins from just attaching to the inner lumen, which can lead to and endothelial cells do a pretty good job at maintaining this sort of Teflon-free environment until the endothelial cells become dysfunctional. And when they become dysfunctional, suddenly, instead of being cellular Teflon, they're like cellular Velcro. And suddenly, everything starts attaching to this inner layer of blood vessels, leading to, the formation of these luminal lesions that can lead to everything downstream from atherosclerosis, thrombosis, inflammation. Now, being that we are a tissue engineering lab, we want to regenerate new tissues. Endothelial cells also play a really important role in tissue engineering because endothelial cells are the cells that make up the earliest or the most primitive blood vessels in the body through a process known as angiogenesis. And in order to make scalable tissues that become increasingly thicker in size, creating a vascularized construct is pretty much critical for the scalability of engineered tissues. And so that's why our lab is very much interested in studying the role of the extracellular matrix. When we talk about the extracellular matrix, what we're referring to is this sort of scaffolding environment or structure that gives cells in our body and tissues in our body 3D shape. But it's appreciated extracellular matrix is not just like a static, scaffolding structure alone, But actually, the ECM is quite dynamic, and it plays a really dynamic role in signaling to endothelial cells as well as to all cell types. Lots of different cues that tell cells when to migrate, when to proliferate, when to die, and what kind of signaling pathways they should turn on. And the way the extracellular matrix does this is through the various properties of the ECM. And these properties can include things like the chemical composition of the ECM, the spatial patterning of the ECM, the stiffness of the material, as well as how the ECM interacts with other kinds of mechanical cues, such as shear or stretch. And so today, what I wanted to do was to focus on one particular focus area. But let me just broadly first tell you, our global research focus of our lab. And so, as I mentioned earlier, we are really interested in studying the ECM cues. And so we can use different techniques, engineer the ECM such that we can focus on those specific characteristics, like their chemical composition using like an extracellular matrix microarray, their spatial patterning using patterning tools like electrospinning or soft lithography, the stiffness of the underlying material using stiffness hydrogels. And using these kinds of ECM cues, we can study the biology of these endothelial cells in terms of their organization, whether or not they're changing their fate, and hopefully apply these for tissue engineering. We are a highly translational lab, and we like to take our in vitro engineered tissues into animal models so that we can study whether or not these engineered tissues are working. And so, we're really excited that some of our engineered tissue work has even gone on to clinics, where we have a biomaterials for treating a vascular disease associated with the lymphatic vasculature, known as lymphedema, clinical testing right now. So for today, what I want to do was to talk about a couple of translational applications of the extracellular matrix for enhancing angiogenesis, that process of creating new vasculature. And for some of these examples, we're using diseases, vasculature that are found both inside the heart as well as outside the heart. But for some of our animal studies, it's easier to see these diseases outside of the heart, but a lot of the implications also apply for within the heart as well. And so today I'll be giving you 3 different vignettes. One that's related to a cell therapy application, another that's a gene therapy application, and a third is a tissue engineering application that all involves extracellular matrix and how we can induce angiogenesis, that formation of new vasculature. So let me start off with a story about cell therapy. So you'll be learning from the other speakers of this month that heart disease is a major killer in the US. And in the case of like heart failure, characterized by the blood vessels in the heart become obstructed, thereby inhibiting the blood flow to the heart. And that's when you get something commonly called as a heart attack or myocardial infarction. But this kind of process occurs outside of the heart. In particular, it occurs this narrowing that occurs in the arms and legs. It's known as another disease called peripheral artery disease, PAD. And PAD is perhaps a less well-known disease compared to heart failure. But PAD is actually very prevalent in the US. It affects over 8 million people, and it's common in those who are over the age of 65. And it's also characterized by endothelial dysfunction that leads to these obstructions that occur in the arm and leg. And the approach is to try to treat these kinds of diseases that are characterized by obstructions is to create or to restore blood flow or blood perfusion to the site of obstruction. And over the years, there have been a number of clinical trials using adult stem cells, meaning cells that include like mesenchymal stem cells, bone marrow-derived mononuclear cells, as examples, and that are injected into these sites of obstruction, they have a problem that they really only have moderate benefit in terms of creating new blood flow or creating new vasculature. And one term that I'll be using today in my talk is the term called tissue ischemia. When blood flow cannot reach the target tissue because of these obstructions, the tissue becomes what we call ischemic. So meaning lacking in sufficient nutrients from the blood and lacking in sufficient oxygen because of these obstructed blood flows. So we can call this as tissue ischemia. And so you guys have probably learned about these induced stem cells, we call them iPSCs, that are essentially our cells that are reprogrammed from any kind of cell in the body, including fibroblasts or cells found in the blood. But basically, they are reprogrammed from the same patient, so we call it autologous. And those iPSCs are very similar to its cousin, embryonic stem cells, in terms of being able to efficiently give rise to endothelial cells, which we can then use for cell transplantation or for tissue engineering applications. So we were excited with the discovery of these iPSCs, and we were the first to create these iPSC-derived endothelial cells for testing in this peripheral artery disease model. And to make it easy for us to track which of the cells that we injected into these animal models, we label them genetically using green fluorescence protein from the jellyfish so that they are fluorescently green, as well as firefly difference from the firefly, so that we can essentially track these cells using what's called bioluminescence imaging. And these cells are very similar to native endothelial cells in that they express no markers of endothelial cells like VE-cadherin and CD31 as examples of markers that are expressed in endothelial cells. But importantly, because the cell density scales linearly with the actual bioluminescence signal, we can use this strategy actually to quantitate the relative abundance of cells in vivo. And so that ends up being a very powerful technique to be able to do this in a non-invasive manner. So to study peripheral artery disease in animals, we utilize a mouse model of peripheral artery disease. It's not exactly perfect, but it imitates the salient characteristics of PAD. In particular, to mimic that loss of blood flow down the leg because of an obstruction, we actually mimic it by removing one of the main arteries by blood flow down the leg, known as the femoral artery, which is in the thigh region. And when we remove the femoral artery in the mice in just one leg, namely in this one leg alone, immediately it reduces blood flow down to almost 10%. Normal blood perfusion capacity. This is shown using a imaging approach called laser Doppler spectroscopy, where the regions that have a lot of colors that are red indicate, high perfusion, but the regions that are really dark here indicate very low levels of perfusion. And of course, in all of the animal studies that we'll be talking about today, they all get all of the approvals from our animal care and use committees to make sure that we're doing everything in as ethical manners. So anyway, using this model of peripheral artery disease in mice, we then injected these iPSCs into that leg that was ischemic, meaning having the obstructed blood flow. And because we can use bioluminescence imaging, we can visualize where the transplant are. And what we found was, hey, the cells are there, but for some reason, cells kind of die off over time, right? That's why their cell numbers decline over time. And in the end, in order to actually have a positive benefit on improving blood perfusion, we actually needed to give 2 doses of the cell. 500,000 on day 0, and then another 500,000 a week later. And even so, there was still a decline in cell survival over time. And so when we found there was such a decline in cell survival, that's when we started to think, can we utilize the extracellular matrix to modulate their survival? And in particular, we utilized a clue from the extracellular matrix known as their spatial patterning. And in a few slides, I'll talk about why we chose spatial patterning among the. But essentially, we first asked, can we use spatially patterned scaffolds as a way to improve their survival in the lake? And we utilized a facile process that was in collaboration with an industry partner, where these scaffolds are essentially made of collagen, an extracellular matrix protein that is found prevalent throughout our bodies, the native naturally derived biomaterial. And we utilize a shear-mediated process that can create nano-patterned collagen scaffolds where their fibrils, these are the collagen fibrils as shown here by atomic force microscopy, are very fine. They actually have on the order of about 50 nanometers full diameters. And essentially they're like little noodles that conglomerate together into a long, thin-like, almost like suture or floss material kind of dimensions, very long and thin strips or strands, you may call it, that are characterized by these highly aligned, or we call it anisotropic, nanofibrillar organization. And the reason for these nanofibrillar organization to be really important is because that's the scale by which cells sense their environment, is at the nanoscale. And they can sense the topography at the nanoscale, and in doing so, align their cell bodies along, essentially like train tracks, so to speak, align their cell bodies along the direction of those nanofibrils. And what you get then are these endothelial cells that are highly elongated. And, and this elongated structure, as seen as scanning electron microscopy, actually very much mimics how endothelial cells look in this very highly elongated morphology along the direction of blood flow in vivo. And so that was the reason why we chose to study spatial patterning, because we wanted to mimic how endothelial cells naturally look at regions of essentially very happy endothelial cells. Those are regions where the endothelial cells produce a lot of nitric oxide, and turns out that they also have this highly elongated cellular morphology. And to show whether or not the spatial topography influences endothelial cell function in terms of their angiogenesis capacity, what we did was that we grew endothelial cells on the surface of the scaffold, and then implanted that cell-seeded scaffold into a 3D collagen gel, and over time quantify the endothelial cell outgrowth as an indicator of angiogenesis, right? Their ability to grow out. And these cellular outgrowths were markedly higher when they were growing on these spatially aligned collagen scaffolds, whereas in the control collagen scaffolds that lack any kind of spatial patterning, the degree of cellular outgrowth was reduced. And when we tried to interrogate what are the molecular signaling that are at play, we first looked at different integrin subunits, because the integrins are essentially these transmembrane proteins on the surface of the cells that basically link the cell to the extracellular matrix. They're essentially the link. So it makes sense to first look at, are their integrin subunits different? And it turns out that the integrin alpha-1 subunit is significantly upregulated at the gene expression level on the aligned, when endothelial cells are cultured on the aligned substrates. And if we try to block that using a pharmacological inhibitor, in this case using a neutralizing antibody, we find that their ability to have this outgrowth is remarkably stunted. So it's suggesting that the alpha-1 subunit is working at play in mediating how endothelial cells are able to sense this spatially patterned topography. But after this in vitro characterization, we went to sort of the main translational study, which was to see if, instead of just injecting cells alone, which we typically would just do using saline, if the cells were preceded onto these aligned nanofibrillar collagen scaffolds, would we get improved survival? And so we hypothesized that these scaffolds are able to provide these physical patterning cues to guide endothelial cell assembly so that they could essentially bridge this area of the thigh that is having tissue ischemia so as to improve the formation of new vasculature. And so we looked at a couple of different groups, and the important ones were initially these studies were done in primary endothelial cells. So these are primary human endothelial cells cultured on aligned collagen or on the non-patterned collagen. And of course, we also have additional reference groups like just cells and saline, our standard approach, or just an acellular scaffold, or just a sham, saline injections. And using bioluminescence imaging, we can see that over the course of the 2 weeks, the endothelial cells that had the highest degree of cell signal were the cells that were cultured on the aligned collagen scaffold. Whereas, as we had expected, that if we just inject cells in saline, they pretty much go away within a few days. And this was surprisingly also true when they were cultured on the non-patterned collagen scaffold as well. And in part because of the fact that these cells were able to survive longer on these aligned collagen scaffolds, we were able to get significantly higher levels of blood perfusion recovery. And the blood perfusion recovery, which is assessed by laser Doppler, the way we quantify that is that, because the contralateral or the other leg is not operated on, we're able to essentially measure what we call the mean perfusion ratio. So we can quantify the perfusion of ischemic leg normalized to its unoperated control leg and get essentially a ratio from that. And so the idea is that if it approaches 1, then that means that it's almost fully restored their blood perfusion. We weren't quite there yet, but made a significant impact in improving blood perfusion using these aligned nanofibrillar scaffolds. And while that work was done using primary endothelial cells, we later showed that this also holds true using the iPSC-derived endothelial cells. And here we utilize a kind of fancy technology that we had developed along with Hanjie Dai, a collaborator in the Department of Chemistry at Stanford, where he had developed these essentially vascular contrast dyes that can be injected into the bloodstream, but interestingly are derived from, these are carbon nanotube ones that actually have very cool optical properties in which they intrinsically emit at very high wavelengths, in this case, 1400 nanometers in wavelength, where in our biological tissues, we really don't have very much native autofluorescence at that kind of emission wavelength. And so by injecting these contrast dyes into the bloodstream through the tail vein, it's able to essentially light up all of the microvasculature in the animals. And so at a very high resolution. And so we're able to then capture images of the microvasculature in the leg. So this is the ischemic leg, and then this is the unoperated leg. And we're able to also similarly show, if we do a ratio of the number of microvasculature in the ischemic leg divided by the unoperated one, we're able to also show that the iPSEC seated aligned scaffolds had the highest relative vascular density. And I won't, go into the details of this busy slide, but basically we're now at the point of trying to understand at the molecular level what's going on using RNA sequencing. And some of our recent unpublished data shows that if we compare the cells on aligned scaffolds compared to the non-patterned scaffolds, we actually see a very different genetic signature. And we can use things like gene set enrichment pathways to identify what are the different signaling patterns that we see. And what we found was that on the aligned scaffold, the endothelial cells expressed many signaling pathways that are associated with the extracellular matrix, or at least with mechanobiology. And some of the highest, the most prominent categories include those that relate to cell adhesion, cell substrate adhesion, cytoskeletal organization, focal adhesions. And this is giving us now these initial clues of how this sort of spatially patterned collagen scaffold is mediating the process of endothelial cell survival and their pro-angiogenic capacities. Okay, so with that work, I wanted to also mention that these spatially patterned scaffolds are not only used just for the application of peripheral artery disease in our lab, but we've also expanded the use of these spatially patterned scaffolds for a number of other cardiovascular and related diseases. So we've used them for myocardial infarction. We've used them for, as I mentioned before, lymphedema, which is when the lymphatic vessels become obstructed. And this is an example of what happens when you have obstructed lymphatic blood flow, where essentially you have a lot of fluid accumulation in the tissues. We've also applied this for related applications to skeletal muscle injury, as well as to the related application of gene and protein delivery. Okay, well, we've talked about how we've used spatially patterned biomaterials for cell therapy, and I want to shift over to some of the recent work we've been doing with gene therapy, which I think is very timely with the vaccines that are developed for COVID. So as you all know, COVID vaccines are developed from mRNAs. And actually, prior to mRNAs were popularized by COVID vaccines, our lab was among the first to utilize mRNAs as a therapeutic strategy for improving angiogenesis. And we had developed approaches to essentially deliver mRNAs from those collagen scaffolds. And some of our initial work, where we were at that time using mRNA encoding just for GFP, so that we can at least visualize when the cells were actually responding to that mRNA by essentially turning on their GFP signaling, such that we can see these green fluorescent protein colors in the cells. And what we found that was really interesting was that when cells were cultured on the scaffold versus the case where if we routinely grow cells on just our tissue culture dishes that are made out of polystyrene, we found that actually the transfection rate was actually much higher on scaffold compared to on polystyrene. And that at the time was shown using just GFP, which is not really therapeutic. So we thought, okay, what if we do this for an mRNA encoding an angiogenic growth factor like HGF, hepatocyte growth factor, which is an angiogenic growth factor. And this is shown here using immunofluorescence staining of HGF. And again, we found that there was a much higher transfection rate on scaffold, on our aligned nanofibrillar collagen scaffold, compared to a conventional case of just using polystyrene. And so to determine whether or not this sort of mRNA delivery from scaffold approach is going to work or not, we initially did some pilot studies where we just implanted collagen scaffolds into the belly of a muscle group on the leg known as the tibialis anterior muscle, which is right below the kneecap. And what we found was, we were able to get a signal. In this case, this is the signal for firefly luciferase using bioluminescence imaging, showing that, in vivo, the mRNA was able to transfect the cells, which led to their ability to become picked up by bioluminescence imaging. And similarly, when we delivered the mRNA from scaffolds that were releasing GFP, we were able to then euthanize the animals and perform histological analysis of the nearby tissue, and again show that there was also the expression of GFP in the native muscle tissue adjacent to the scaffold implant. Before we talk about some of our large animal studies, I want to take a step back to mention why we're interested in mRNA in the first place, compared to other kinds of techniques. The reason why we're interested in mRNA as a therapeutic is because of the fact that we know mRNAs, unlike things like other kinds of gene delivery, like DNA, for example, mRNA cannot integrate into the genome. So just from that, we know that mRNA therapies are much more safe because they cannot integrate into the genome. But mRNAs are also somewhat transient. They are rather short-lived. They only last on the order of days to weeks. And so, or in the case of the COVID vaccine, maybe a bit longer, but in the end, they are a bit, transient. And this is also beneficial in the case of when we want to give a therapy that is not sustained forever. Now, you can imagine if we were giving angiogenic drugs that essentially create new blood vessels all the time, that could be bad. You can get something, the overgrowth of blood vessels that can lead to very leaky leaky blood vessels, which are not good for the body. And so, our therapies to improve angiogenesis should not be something that is sustained forever. And so we do want something short-lived. And that's another good property about these mRNAs. Okay, so that's the reason why we chose to use mRNAs in the first place. Because our lab is a very translational lab, and we want to see how we can scale our technology into animals that are closer to resembling humans, so we have a closer idea of how humans would respond to it. We use, in this case, a pig model of PAD. Now, pigs are much bigger in size compared to mice, and we had to figure out a way to adapt our technology from mice to pigs. And we utilized this sort of hybrid approach where we still had our nanopattern collagen strip in the inside, but essentially wrapped almost like a cinnamon bun by multiple layers of sheets of collagen that are impregnated with HGF mRNA, which is shown here by the green color, creating essentially this structure that looks like a much thicker scaffold that has HGF mRNA inside it. And so when we implanted, actually, before we jump to the in vivo study, I forgot to mention that we did perform some characterization to indicate what is its release kinetics of the mRNA. And so using our quantitative analysis, we're able to confirm that the release of the mRNA actually looks like there's a burst effect initially, followed by a slow, gradual, cumulative, a release over the course of a month. And initially, we were really kind of, disappointed by that. But what was intriguing was that despite having this initial burst effect, which was not what we were hoping for, but in terms of what is the outcome, in terms of the cells getting transfected with HGF and the ability for the cells to continuously produce HGF protein, this is shown here by ELISA analysis of HGF protein, we found that the cells were actually able to have sustained elevated secretion of HGF protein over the course of 30 days compared to the dotted line, which is the endogenous level of HGF otherwise. So even though the release was more of a burst release kind of kinetic, but we were able to get sustained release of the protein after the cells were successfully transfected. Okay, so then we were much more enthusiastic about this pig study. Pigs are very expensive to work with. So this was initially, I would say, more of a pilot study of 5 pigs, where we, in one leg, implanted the HGF scaffold, and in the other leg, actually implanted nothing. So it was just a control leg. So both legs, in this case, received ligation of the femoral artery. So you can see those are 2, with the clips over there, that's the femoral artery. And then we implanted the scaffold adjacent to the site of this ligated femoral artery, hoping that, again, the scaffold can bridge that area of vascular obstruction. But what was really interesting was that when we followed the animals using CT angiography over the course of 5 weeks, we were able to show that over the course of 5 weeks, there was a difference and a benefit of the HGF releasing scaffold. In particular, near the site of where the scaffold was implanted, we're able to visualize more, the formation of more vasculature in that HGF scaffold implanted leg compared to that of the control leg. And then we're able to follow up to do histological analysis to visualize the formation of new vasculature at a microscale level. And we're able to show that as we expect, the mRNA is being released locally to the scaffold, which means that the improvement or the formation of new vasculature is largely going to be confined to local regions from where the scaffold is. And so we quantify the formation of new microvasculature as a function of different radial distances from the edge of the scaffold. And so we found that there was a significantly higher degree of microvascular formation within 50 microns from the outer edge of the scaffold, compared to if you look out at much further distances. So this is really showing us that the mRNA is able to act in a spatial defined way where they really only act on the cells local to the scaffold environment as we expect. And that was also one of the benefits of delivering these mRNAs from scaffolds, so that the effect would be localized only to the region adjacent to where the scaffold is implanted. So what we think is going on, based on, granted, this pilot pig study, which we are continuing to work on to expand on, but what is going on in the system is that when we deliver these implanted scaffolds that have the mRNA of HGF, the mRNA transfects local cells, and these local cells can include things like fibroblasts or muscle cells, for example, that are adjacent to the site of implantation, and then that these scaffolds, transfect these, fibroblasts or muscle cells, and those cells then become transfected. They produce HGF protein, which then recruit the endothelial cells, which are shown here by the yellow cells. And then those endothelial cells, because they're recruited, are able to form new capillary growth along the scaffold over time. And so this is our working hypothesis of what these scaffolds may be doing over time. Okay, well, I've talked a little bit about cell therapy and gene therapy, and one of the last applications I'll talk about is a tissue engineering one. Now, even though we are a cardiovascular lab and we have developed many kinds of engineered tissues, including engineered cardiac tissue, for this one, I want to give an example that's actually related to skeletal muscle, because the idea is that the engineered muscles that we make, whether skeletal muscle versus cardiac muscle, all share a lot of similarities. But because I'm also at the Veterans Affairs Hospital, we also try to work on research that is relevant to the veteran population. And a lot of the insights that we learn from these skeletal muscle applications, we are already applying into the cardiac muscle application as well. But with the slight advantage in that, in the case of muscle, skeletal muscle rather, the animals don't survive much better, right? Whereas in the case of when we work on the cardiac muscle, the disease model is so severe that many of the animals actually end up dying. So we actually like to study a lot of the proof of concept in skeletal muscle first before we transition them into the cardiac muscle. So that's another reason why we started some of our initial work in skeletal muscle, where the goal was to create a vascularized muscle. And as I mentioned, to create any kind of engineered tissue that's scalable, we need to be able to create a form of blood flow or blood perfusion in order for the cells to maintain their ability to gain nutrients. And in the case of skeletal muscle, one of the disease applications that are relevant to our veteran populations is volumetric muscle loss, which is associated with the loss of a significant chunk of muscle due to trauma like traumatic injuries due to gunshot wounds, roadside bombs, things of that nature. And even though skeletal muscle can regenerate generally pretty well, in the case of if a huge chunk of muscle is removed, in that case, that skeletal muscle will not be able to regenerate. And so very similar to cardiac muscle, skeletal muscle can generally be considered as a bundle of muscle fibers that are organized in parallel, interspersed with microvasculature that are also organized in parallel. And so, with this sort of simple geometry, we were trying to recreate this using our spatially patterned collagen scaffolds. And our approach was to mimic these sort of bundles of myofibers. We essentially bundled up our long and thin scaffold strips and bundled them together in parallel, where we seeded them with both skeletal muscle cells, known as skeletal myoblasts, as well as endothelial cells. And when we initially co-cultured these two cell types, the myoblast or the myoblast with endothelial cells, we first wondered, in this co-culture scenario, does it impair the ability for myoblasts to turn into muscle fibers? And it turns out, and we use this myosin heavy chain antibody shown in green to label the formation of these myotubes, which is something really unique to skeletal myoblasts. They fuse together in the process to form these multinucleated myotubes. and which are basically the precursors of muscle fibers. And the endothelial cells actually, not only did they not interfere with the process of making these muscle myofibers, but were able to actually make even longer muscle fibers, as shown here by the myotube lengths. But probably the most striking observation, as we would expect, is that when the cells are growing on the aligned scaffolds, which are shown here maybe in this example better, the cells reorganize and sense those underlying kind of like railroad tracks to essentially cause them to align in parallel, in this case horizontal, because that's the direction of the collagen nanofibrils underneath them, and are creating essentially this almost like a parallel bundle of myofibers. And these parallel bundles of myofibers are then able to become organized and contract synchronously in response to electrical stimulation here. Hopefully you can see from the video, they're able to contract synchronously, which is great for skeletal muscle. We also did some proteomic analysis to identify whether or not the endothelial cells are able to have other benefits. And in particular, one of them is whether or not, co-culturing these myoblasts with endothelial cells are able to have some kind of therapeutic benefit. And to do this, we looked at actually proteomic analysis and found that when endothelial cells are in co-culture with the muscle on the aligned scaffolds, the combination of the two, this co-culture and the aligned scaffold, is able to induce the release of pro-angiogenic cytokines. And using proteomic analysis, we found a couple of examples of some of these pro-angiogenic cytokines like vascular endothelial growth factor and angiogenin, as well as some that are myogenic as well, also meaning actually improving muscle regeneration, such as the insulin-like growth factor binding protein 3. So it's suggesting that these endothelial cells are not only beneficial, perhaps as priming the way to create new vasculature, but also having direct effects on the myoblasts themselves. And so in our animal model, this is what we did. We created, again, our bundle of myofibers and implanted them, in this case, into a mouse model of volumetric muscle loss. The way we do this is that we try to mimic the sort of traumatic, loss of a huge chunk of muscle by surgically excising about 40% of the muscle volume in one of the muscle groups known as the tibialis anterior muscle, again, the one right below the kneecap, and in its place implanted this muscle bundle. And our muscle bundles consisted of those that only had myoblasts, meaning those that only had the myoblasts that were either in a randomly oriented collagen scaffold or an aligned collagen scaffold topography. And similarly, the ones that were co-cultured together, which we call an endothelialized engineered muscle. And when we compared how do these skeletal muscle cells respond after being implanted into this mouse model, we used, again, bioluminescence as a tool to measure cell survival over time, and found, interestingly, that the group that had the highest degree of cell survival as well as proliferation was actually our group here, which is shown by the solid black lines, which is our co-cultured, rather our endothelialized engineered muscle that are growing on the aligned collagen scaffold. But overall, the cells were able to survive, reasonably well in all groups. But it really was until we looked at the tissue histology at 3 weeks after implantation that we really be able to see differences in vivo. Namely, these transplanted muscle fibers were also labeled with GFP in addition to firefly luciferase. So these green-looking cells are our engineered myofibers that have now taken root and formed bona fide tissue muscle fibers that also express myosin heavy chain. And markedly, you can see that on the aligned scaffold delivered constructs, there was a lot more transplant-derived myofibers compared to on scaffolds that were what we call randomly oriented. So this really showed us that the aligned scaffolds are really able to improve the muscle regeneration. But now what happened to the endothelial cells? Remember, we co-cultured the endothelial cells with them. What did the endothelial cells do? And we also did separate experiments where instead of the myoblasts being labeled, the endothelial cells were labeled instead. And we can show over the course of 21 days that the cells didn't really proliferate much, even though they did survive, we can still detect signal after 21 days. But most importantly, what we found was that those endothelial cells were able to contribute to the formation of more perfused vessels. So what we mean by perfused is that at the time of euthanasia, we injected them with a different dye known as isolectin that binds to all of the endothelial cells that are perfusable. And when we can histologically quantify these endothelial cells that dually express isolectin as well as CD31, an endothelial specific marker, we're able to essentially quantify the perfused microvessels in the tissue samples, and found that again, on the Align scaffolds, the number of perfused vessels were significantly higher. So this is showing us that the Align scaffolds not only improve muscle regeneration, but also vascular regeneration. But I think one of the coolest images that I think I'd like to show here is actually this 3D reconstructed confocal image of the tissue in vivo, where what you can see here shown in green are the myofibers that are transplant derived because they're GFP labeled in green. And then the red here is isolectin, which is again visualizing the perfused vessels. And what's really remarkable is if you look here, you can see that the myofibers are kind of all aligned in parallel, and the vasculature is also aligned in parallel. So this is pretty much mimicking what we are seeing in the normal physiological environment, right? Where the muscle fibers are again parallel bundles interspersed with vasculature organized in parallel. So this was really cool because it was showing us that the spatial patterning alone was able to better recapitulate this sort of more native looking muscle regeneration process. Whereas when we looked at the randomly oriented scaffold, this is just looking at the isolectin, and you can see that the formation of the vasculature is a lot less organized. And these insights that we're gaining from skeletal muscle, we've also recently applied to cardiac muscle, where we are able to create highly organized skeletal muscle constructs as well. And so, I know that we're running low on time, but I just wanted, to throw in a plug there of some of the cool things that you can do, maybe as a motivational factor for many of the trainees there, that sometimes, science has, its mysterious ways. And we were able to apply this technology for actually doing work in space on the International Space Station, where we took this concept of creating engineered muscle to model a muscle aging disease known as sarcopenia that happens just with the process of aging. And we were able to get funding to send our engineered muscles into space to see how in space, how the microgravity itself can be accelerating that process of aging, including muscle aging. And this is also based on what is known from many of the astronauts who go up into space, where, when they come down, their body has effectively aged by like a decade or something. And so we were really happy that our engineered samples were sent off into just the past year. And now, we just got those samples retrieved back where the astronauts, did some of the experiments for us, where we are now looking at the effects of different kinds of drugs to see if the drugs can reverse that process of muscle aging. So anyway, so this is, some of the cool work that, that we can do that sometimes is, really exciting and also having, benefit for biomedical research. Well, I know that I don't have, enough time to go through some of the other work that I wanted to talk about, but I'm thinking maybe I can essentially summarize some of the work here to say that, hopefully today in these 3 different vignettes, I was able to talk about some of how spatial patterning is a really important ECM cue that can do many different things for the endothelial cells of our vasculature in terms of improving their survival, their angiogenic capacity. And we can also use it as a tool to deliver drugs like mRNAs that are encoding angiogenic growth factors. And ultimately, in the last example, although I was using skeletal muscle as an example, we're able to know that we can use these spatially patterned scaffolds to create organized, vascularized muscle tissue, which we are now applying in our cardiac muscle research group to do the same thing. And so with that, I'd like to acknowledge my lab members who did a lot of this work, and I'd like to acknowledge our funding sources. And feel free to email me here if you have questions or interest in postdoc fellowships. And with that, I would be happy to take your questions.
Well, so we have about, I don't know, 20 people here in the room, and it looks like we had about 60 people online, so you had a pretty good audience today. So we can take questions. I think there might be some in the chat or Q&A, and then people in the room can think about them as well. So it looks like we had one from Daniel Chaparro in the chat, who asked if scaffold treatment or sort of perfusion, also angiogenesis, creates a non-physical microvasculature compared to uncompromised legs. Will creating all these new branches and bifurcations have negative effects on the vasculature and surrounding tissue, and would it change cardiac afterload?
Sure. So, I think we are probably, I would say, at the first stage, right? So in the case of the heart or in the case of the peripheral vasculature, where you experience tissue ischemia, the first priority is to improve or to restore blood flow and blood pressure, right? And then the second priority would be, okay, how can we create this restoration of new vasculature that mimics the native physiological history? So in our case, we were trying to do a little bit of that, and admittedly, our, spatially patterned scaffolds are not complex enough to truly recapitulate the full topography organization of microvasculature, whether in the heart or in the skeletal muscle. But I think it served its primary purpose, which is to do the first priority, right? To first get more blood flow, get more oxygen, so that there's less ischemia. Of course, if there is a concern where basically where the newly formed vessels do not recapitulate the native structure, yes, it's possible that can have some other adverse consequences. But actually, if you think about a lot of the existing clinical trials and where they've used angiogenic growth factors in the form of proteins, they've also used cell types, and all of those therapies that have gone through the trials, are they really able to control the formation of the vasculature down to intricate details? No. And the idea is that the host is able to essentially remodel the vasculature to some extent as well, according to what the body needs. And I think it's the same idea for us. We're not fully recapitulating, the 100% the microvasculature organization, but the idea is that in vivo, the body is able to intervene and make course corrections as necessary.
Great, thank you. Other questions here in the room? So, yeah, I know. What?
For these scaffolds that were used for mRNA transfection, how was the mRNA molecule functionalized into the scaffold?
Yes, so that's a very good question. So I didn't go into a lot of details into that. So let me clarify. So when we were trying to develop a way to deliver mRNA, we were considering, our design considerations. We wanted something that was simple, so that it wouldn't have like complex chemistries, because ultimately we wanted something with near-term clinical potential. So we didn't want to introduce like cross linkers with lots of strange chemicals. So we ended up using something that was highly facile. So in particular, what we actually utilized was pretty much a freeze drying approach to essentially use negative pressure to essentially pull the mRNA into the 3D scaffold. There was no covalent conjugation. There was no cross-linking involved. It was purely based on essentially freeze drying and the negative pressure associated with freeze drying to essentially suck the mRNA into the scaffold. And that was probably by, not surprisingly, the release kinetics showed a burst over time because there was no covalent conjugation. And of course, we could have used like more sophisticated chemistry, but another design criteria is that even though mRNAs that we're using now are chemically modified in a way to be relatively more stable, in the end, they're actually not. So we were worried that manipulating mRNA into different covalent strategies would lead to the loss of bioactivity. So that was another reason why we ended up using something very easy that doesn't really manipulate the mRNA too much.
Thank you.
I see two more in the chat. I think the first one is on how close you were able to recover, post-introduction to the scaffold, how closely you're able to recover oxygenation and function after ischemia. So I know you shared a little bit of data on that. Maybe you can speak to that a little bit more.
Sure. I think it's related to some of our peripheral artery disease work. And what we directly measure as the output is blood perfusion using the laser Doppler spectroscopy method. And so when we quantified what we call that mean perfusion recovery, which was the perfusion of the ischemic leg divided by that of the unoperated lateral leg, that is actually our functional readout, because that's what we really want, is to improve the blood perfusion. Now, we don't exactly measure the oxygenation per se, but the implication is that getting more blood, which is oxygenated, then the oxygen level must be also getting closer back to normal.
And then the last question here, have you looked into any inflammation or immune responses that occur after implantation?
Yeah, so that's a very good question. I didn't have enough time to go through that because most of our studies, at least in the peripheral artery disease context, is at about 2 weeks after implantation. In the case of our skeletal muscle work, we looked a little later at 3 weeks. But in these several time points, a lot of the acute inflammation is gone, right? So when you create an injury or disease, there is initially this phase known as acute inflammation. And so for most of our time points, we've already gone beyond inflammation. So at least histologically, we don't see marked levels of inflammation, although they are sprinkled about, but nothing very significant. The other thing I want to point out is that in the case of our peripheral artery disease models, we look at human cells, right? And they're delivering human cells into mice. The mice by default have to be immune compromised, otherwise they would reject all of the cells. And so we have been using immune compromised skin mice for that study. And because of that, their immune system is actually not completely normal. So that's also another caveat of our human, which is that we cannot study them in a fully immune animal. But related to this, we have done some in a previous work where we used mouse cells. In this case, they were embryonic stem cell derived endothelial cells. And because they were mouse-derived, we were able to generate them from the same strain of mice we call syngeneic cells and implant them back into the same strain of mice that were immune competent. And in those studies, we also did not observe marked inflammation histologically at 2 weeks. So we think that our cells are safe and not eliciting, terrible amounts of inflammation.
Thank you so much. I want to remind everyone before we close that if you're a grad student, you're certainly welcome for us to have lunch. Dr. Wong will be having breakfast, I assume, at noon, and that'll still be in 2680, so she'll be broadcast there. You can meet with her and ask more questions. And she's got a busy day ahead. So let's thank Dr. Wong again.
Thank you very much.